Hello, everybody. Today, let's move this. All right, notes for today. I'm going to start off with talking about cell size. All right, so topic 2.3 is cell size. Our learning objectives explain the effect of surface area to volume ratios on the exchange of materials between cells or organisms in the environment, and explain how specialized structures and strategies are used for the efficient exchange of molecules to the environment. So with our eukaryotic and prokaryotic cells and their cell size, a couple of important structures involved with that exchange of materials with the environment. We have the cytosol, which is the fluid in which all of our organelles are found. Commonly, it's referred to as the cytoplasm. That refers to this region. And then there are the membranes. In order for something to diffuse into a cell, especially a simple cell like a prokaryote, they have to pass through the cell membrane. Then they go into the cytosol where they can diffuse to where they are needed. In the case of the bacterium or the prokaryote, they might head to the ribosome or to any of the reactions occurring within the cytosol. In case of a eukaryotic cell, things like oxygen will flow to the mitochondria. In a eukaryotic cell, there are plenty of membranes like the endoplasmic reticulum and membrane-bound organelles, such as chloroplasts and mitochondria, that help to localize or compartmentalize different processes, and we'll get to that in a few minutes. Comparing our two types of cells, both types have a plasma membrane or a cell membrane, as well as ribosomes. The eukaryotic cells, however, do not have the membrane-bound organelles, and they don't have a nucleus. The shape of their DNA is slightly different as well. In the case of a prokaryotic cell, the DNA is going to be one circular molecule. And in the picture, it looks like a squiggle because it's just sort of been folded up on itself. Eukaryotic cells, their DNA is linear. So depending on the type of eukaryotic cell, there are going to be varying numbers of pieces of DNA. In the case of a human, the normal number to have in a human would be to have 46 pieces of DNA. We call those pieces the chromosomes. So now cell size plays a big role in the ability of the cell to get those materials in through their membrane and transport them somewhere. So our cells tend to be very small, and the reason why our cells are small is because of their surface area to volume ratio. As the volume increases, like we see over here, the surface area to volume ratio decreases and the movement of materials becomes less efficient. The surface area of the plasma membrane or the cell membrane might be large, but it also needs to be able to adequately exchange materials. Smaller cells are going to have a more favorable area to volume ratio for the exchange with the environment. Different metabolic requirements also impose limits on the size of the cell. So if the cell needs oxygen and there's not enough around, it's not going to grow that big. Eukaryotic cells can become bigger because they have those organelles. And those organelles allow our eukaryotic cells to specialize. So let's go look at, oops, I don't have them open yet. Note set 210 on compartmentalization, which is the specialization. And so eukaryotes, we'd said, have some special membrane-bound structures. We're going to look at just 3 of those and look at how their internal membranes and organelles contribute to this compartmentalization of our eukaryotic cell. So here are 2 typical eukaryotic cells. We have animal cells and a plant cell. Keep in mind, eukaryotes also include fungal cells and protist cells, but these are the 2 that usually show up. And we can see in these cells lots of different organelles. We have perioxysomes, the Golgi apparatus or Golgi body, vesicles, mitochondria. We have an ER or the endoplasmic reticulum, the nucleus, some ribosomes. Over in the plant cell, we also have chloroplasts. We've got vacuoles, lots of different organelles. However, when it comes to internal membranes, the mitochondria and the chloroplast both have their own internal membrane. If we look up here at the top right, this is showing the internal membrane of a mitochondria. So here's the membrane of the mitochondria. And then in here, this single strand, that's the inner membrane. This inner membrane is very important. It breaks up the 3 reactions that we need for cellular respiration into 3 places, allowing things to run more efficiently. First part of cellular respiration actually occurs in the cytosol outside of the mitochondria. Then there's the Krebs cycle and the electron transport chain. By splitting these things up, it's now allowing, when we talk about cellular respiration in more detail later, we're going to learn there's pH differences that are required for these reactions. There's also enzymes required. And this inner surface of the mitochondria is folded up on itself. That gives a lot more surface area to have a lot more enzymes that are necessary for this reaction. And if there's a lot more enzymes, then this reaction can be happening at a lot of different places within the mitochondria. We go back here for a minute. In our cell, we can see here the mitochondria, lots of different little wiggles and squiggles. That's all the folded inner membrane, increasing the surface area. Chloroplasts also have a folded membrane inside. It's harder to see here in this picture. It tends to look like little green pancakes in a diagram. But that membrane is called the thylakoid membrane. Important for breaking up the parts of photosynthesis and all the different reactions involved. Also, same reason as a mitochondria, it's all folded up like that to give increased surface area for the enzymes in a very small amount of space. So the volume is very low. And it's that surface area to volume ratio. Another example we have is the endoplasmic reticulum. This organelle is a set of folded membranes. In this case, the increased folding gives us increased surface area for more ribosomes to attach. You might remember from prior biology classes that there's the rough ER that has ribosomes attached. The more ribosomes there are, the more protein synthesis that can occur, because ribosomes are important for protein synthesis. We can also see in this diagram, let me move that around, that the endoplasmic reticulum is very close to the nucleus, so that when the mRNA leaves, it's not having to find ribosomes. It's also relatively close to the Golgi apparatus, which plays an important role in packaging any protein that's going to have to exit the cell. And so this, which Golgi also has, folded membranes, increasing surface area. So now you might be wondering, how did we get these special organelles? Which brings us to topic 211, the endosymbiotic theory. So we've got to describe the similarities and differences in compartmentalization between prokaryotic and eukaryotic cells, and then described the relationship between the functions of the endosymbiotic organelles and their free-living ancestral counterparts. So eukaryotes have a lot of compartmentalization. Really struggling with that word today. Prokaryotes don't. Enzymes that they need for photosynthesis or cellular respiration are all going to be on their outer plasma membrane. But those molecules, those prokaryotes and those enzymes on their outer cell membrane did give rise to our mitochondria and our chloroplast. Here we've got our endosymbiotic theory is what explains this. This was first proposed by microbiologist Lynn Margulis back in the 60s. Her original hypothesis, because remember in biology we have a hypothesis, then when it gets tested and we get lots of evidence, it goes up to a theory, which is going to be more widely accepted and valid. So she proposed mitochondria are the result of endocytosis of aerobic bacteria. Chloroplasts are the result of endocytosis of photosynthetic bacteria, both of which, in both cases, the endocytosis is, we will refer to it as cell eating. In both cases, they were taken in then by a larger anaerobic bacteria who would not otherwise be able to exist in an aerobic environment. And she proposed that the arrangement became mutually beneficial for both cells. Couple of pieces of evidence she had for this. One was size. When we look at the size of a typical prokaryotic cell and the size of a mitochondria or a chloroplast, they are both very similar, and they're similar in size to each other. We can look at their DNA. Mitochondria and chloroplasts both have their own DNA, and it is a circular DNA like we find in bacteria. When it comes to how mitochondria and chloroplasts reproduce and make more within the cell, they reproduce through a process that looks very similar to binary fusion, which is the process by which prokaryotes divide, and it's more different than looking at how some of our other cell organelles are formed, like the endoplasmic reticulum, the cells that's going to build more membrane. The final thing was that these ones, the mitochondria and the chloroplasts, have two membranes. They have an outer membrane and then that folded inner membrane. Where did that come from? There we are. So that membrane, the result of our double membrane organelle, came from the process of endocytosis. So in this case, so here's an example. Let's move that so we can see our picture. Here's our host cell. It's engulfing a bacterium. And so now we have the vesicle membrane, the bacterial plasma membrane, and then over time, as these symbiotes became more and more dependent on one another, we now have the outer membrane of the mitochondria or the chloroplast, and then the inner membrane. So our theory states that aerobic bacteria that require oxygen were ingested by anaerobic bacteria that would otherwise be poisoned by oxygen. And they each have a survival advantage as long as they continued their partnership. The result of this engulfing is a cell with a double membrane bound organelle. So the inner lipid bilayer would have been the cell's plasma membrane, and the outer lipid bilayer came from the cell that engulfed it.